On to chapter 2. Well, we actually went to chapter 3, now we're going back to chapter 2. Traditionally, genetics is taught from a Mendelian perspective, showing how the genes segregate during meiosis, how independent assortment works, and how that affects the outcome of genetic crosses. And in this case, and then to explain how all that works, how you get all these patterns, how you get your 1 to 2 to 1, 3 to 1, 9 to 3 to 3 to 1, you need to look at the chromosomes and see how the chromosomes are dividing during meiosis. And so this chapter is all about the chromosomes and how they divide, and that explains pretty much everything you need to know about genetics. That might be a little bit of an overstatement, but it has been said by many very good geneticists that if there's one thing that students should understand in order to really get a handle on genetics and to really have a good understanding of it rather than memorizing various ratios, if you understand meiosis, it really explains everything. And meiosis is a hard one to get sometimes. And the only way to do it, in my mind, the only way I can do it, is to draw out the chromosomes. When I was in graduate school, I thought I was pretty good at genetics going in. One of the first meetings I had with my PhD advisor, I was presenting a paper, and he wanted me to explain it in more detail. He said, draw out meiosis. I had no clue how to draw out meiosis. That was a little traumatizing, so maybe that's why I insist on my students being able to draw out meiosis. That and because it's super important in genetics. So we are going to talk about cell division. And of course, there's two types of cell division, mitosis and meiosis, in eukaryotic cells. This is a picture of a cell dividing, presumably by mitosis, because that's usually what the pictures are. It's just easier to get mitotic cells. And the blue things here are chromosomes. The green things are spindle fibers. Now the red, it's probably actin or something like that. Spindle fibers are these guys that are made up of microtubules, and they're latching onto these chromosomes and they're pulling them apart. It's a very cool picture. We'll have to take a look at some videos. So this cell is probably in metaphase. The chromosomes, you can actually see them. They're all scrunched up, and it's just a cool picture. Now, the opening story has to do with this blind man's riddle and people buying socks and blind man with socks. I don't really get it, so I wouldn't worry about that too much. All right, so here, prokaryotes versus eukaryotes. We're just talking about eukaryotes. We're talking about eukaryotes and how they divide. We will come back later to prokaryotes, but there's really not much to prokaryotic cell division. They just copy their DNA and divide. Cell reproduction, mitosis, and sexual reproduction, meiosis. I suppose it's good that we get to talk about sex, but it's not really as exciting as you might expect. So here's prokaryotes versus eukaryotes. DNA, prokaryotes have DNA. Eukaryotes have DNA. Everything has DNA because they're alive. And if you're going to be alive, you need to have DNA. All right, so prokaryotes have DNA. They also have ribosomes. That's something I want you to remember. A lot of people forget that bacteria have ribosomes. Bacteria do your DNA, RNA, protein gig. And then eukaryotes, they have a nucleus, nuclear envelope, endoplasmic reticulum, all these different types of organelles, and they're much more complex. But what we care about most is the nucleus, and inside the nucleus we have chromosomes. And that's what chapter 2 is all about. There's a comparison of prokaryotes and eukaryotes. We don't really care about prokaryotes at this point. So here's a chromosome. Now, this is cool. Here's some DNA. So if it's a chromosome, like this, you can actually see it's a eukaryote. This is sort of at the super microscopic level, the structure of chromosomes. You have DNA. These things are called nucleosomes. And the proteins that make up the nucleosomes are called histones. And we're going to talk about all that stuff in a lot of detail. That's really, really cool and has a lot to do with chromatin structure and how genes are expressed. It's just talking about what the structure of this is, and this is an actual chromosome. We'll come back to that. So we won't be asking about this kind of stuff up here on the test. This is a chromosome, and this is a chromosome that has a centromere here, which is a constriction, telomeres at the ends, and then you have two chromatids. So a chromatid. So this is a chromosome with two chromatids. Chromatid, not to be confused with chromatin, but don't worry about that. We're not worried about that. We're not talking about that for a while. Viruses, we don't care. We just want to talk about meiosis. All right, so eukaryotic cell replication. So eukaryotic chromosomes in sexually reproducing organisms come in homologous pairs. What that means is when you draw a chromosome, each chromosome, well, it can start out like this, and then it becomes like this. That's S phase. We're going to talk about the cell cycle. That's where S phase comes in. And then here's a chromosome, and it has two chromatids. And these chromatids are identical because they just got copied. So if you have a big A on one, you got a big A on the other. So we'll talk about that. We're not going to talk a whole lot about chromosome structure, just a little bit. The cell cycle and genetic consequences of the cell cycle. And that basically means that, so if you think about cells as being diploid, so that means they have two pairs of chromosomes. Like humans, we have 46 chromosomes, 23 pairs. If we do mitosis, our cells have 2n, 46 chromosomes, and when they're done they have 2n, 46 chromosomes. Because mitosis makes identical cells. Meiosis, we start out with 2n, which is 46 chromosomes. We go down to n, which is 23 chromosomes. That's the whole point of meiosis. And because we're talking about genetics and how alleles segregate, how independent assortment occurs, all that kind of stuff, this is our big deal right here, meiosis. So diploid cells carry two sets of genetic information. Okay, that's good. Haploid cells carry one set of genetic information. So you can actually have diploid cells that divide, that's an N, diploid cells that divide by mitosis and make 2n. So if you start out with 2n, you go to 2n. There are some haploid cells, some kind of yeast cells and other kinds of cells that also divide by mitosis, and they start out n and they stay n. No matter what you start out with, you have to be the same after mitosis, because mitosis makes identical cells. But meiosis, see if I can write this on every slide. Meiosis, you go from 2n to 1n. 46 to 23. Let's say you have some other crazy organism that has a 6 chromosome total, you go down to 3. And in meiosis, the chromosomes are in pairs. So here's a human karyotype. Let's see, it doesn't say that word on there, so let's go karyotype. And that's how you look at chromosomes. You'll notice that some in the little constriction here is a centromere, and sometimes it's super towards the end. Like you can, like here, on chromosome 21, this is the infamous Down syndrome chromosome. If you had an extra one, you'd have Down syndrome. But the centromere is right at the end. So sometimes the centromere is at the end, sometimes it's in the middle, sometimes it's somewhere else. These chromosomes are in pairs, so we have 23 pairs. We've got 22 chromosomes that are autosomes. Autosomes means they're not sex chromosomes. And then we got sex chromosomes. In this case, there are 2 X chromosomes, and so this is a female. So that's the kind of info you can get from a karyotype. But if you have a chromosome, you can draw them like this. This is a chromosome with 1 chromatid. This is another chromosome with 1 chromatid. And these are homologous pairs, but they're homologous pairs before DNA replication. And so you have a chromosome with a locus, and that locus is the gene A. And here we have one version of gene A, which is allele A. Here's another version of gene A, which is allele little a, big A, little a. Big A is dominant to little a. So that's kind of a convention. If it is given a capital letter, it's dominant over the other one. All right, there's a close-up of that. So let's do some DNA replication. I'm sure it'll do this in a minute. So now we're going to copy this chromosome, and that's how they look before they go through cell division, whether it's mitosis or meiosis. You have to copy the DNA first. And if you copy it, if you had an A here, we got an A here, we got a little a here, and a little a here. So just a little bit about chromosome structure. The centromere is in the middle. When the cell divides, the spindle, I mentioned the spindle a lot. The spindle is a structure made up of microtubules, and when the cell divides, you'll see chromosomes that look like this. So during mitosis, they might look like this. Oops, so that would be getting pulled the other way. So they're getting pulled apart in the cell. You have telomeres. Telomeres are on the tips of the chromosomes. And we have origins of DNA replication. That's the beginning of DNA replication. And we'll talk about that more when we talk about molecular biology and DNA replication and that sort of thing. So there's just some more stuff about chromosomes, different structures, your spindle, microtubules. Kinetochore is sort of this whole structure that's involved in cell division. It has more to do with the mechanics of cell division. I'm more interested in you understanding what the chromosomes do, not exactly how they do it. I mean, I want you to know that in meiosis, the chromosomes line up in pairs, and then homologous chromosome pairs, members of the pair, segregate into different cells. That's in meiosis. In mitosis, the individual chromosomes, the chromatids, split and go into different cells. So that's a big, huge thing, understanding the difference between chromatids and homologous chromosomes. But it's also useful to think about the cell cycle for lots of different reasons. In this case, we're going to talk about in terms of cell division. And M phase can be mitosis or meiosis, depending on the cell cycle. Checkpoints are important. We think of these mainly in terms of mitosis, and we'll talk about those in a minute. But checkpoints are important because they keep the cell cycle under control. If these checkpoints go out of control, so if they go out of control, so a checkpoint is sort of like checking to make sure the cell is ready to divide. If it's not ready to divide and divides anyways, very simplistically, we call that cancer. There's at least a step towards cancer. I'm not going to talk a lot about that right now. We're going to talk a whole lot about it later in the semester. It's very interesting. But here we mainly want to know what's going on with the chromosomes, but we need to understand the cell cycle. So G1 is growth phase or gap phase sometimes. G0 is a non-dividing. That's what's key about that, is that cells can sit there and not divide. And then sometimes you give them a drug or a hormone or something like that, which will kick them into the cell cycle, and they'll start dividing. So a lot of times you have cells that aren't dividing, and that's sort of what sometimes cancer-causing agents will kick them into the cell cycle and tell them to start dividing. And a checkpoint is just sort of a point that if a cell gets past that point, then it continues on through the cell cycle. And like in this phase, you don't want to pass this cell cycle until certain things happen. Or you don't want to pass this checkpoint until certain things happen. For example, you want the cell to get to be a particular size. And you don't want it to divide before it gets to be a particular size, because then cells will get too small. If you G1S, because S is the S phase, S stands for DNA synthesis, so that's what the S in S phase stands for. Quite a bit of stuff that happens during the cell cycle, and as I said, we'll come back to that in quite a bit. But we're basically preparing for DNA synthesis, doing DNA synthesis, preparing for mitosis, and then we get into spindle assembly checkpoint. You want to make sure all the spindles are attached so that when the cell divides, all the chromosomes get pulled where they have to go. You don't want some to be left in the middle and then not get distributed to the right cell. And then cytokinesis is when you have actual cell division. So we're going to talk about the stages of mitosis in a pretty sort of general way. We're not going to look at all the details of everything that happens in each phase. We mainly want to know what's going on with the chromosomes. So you can think about the cell cycle. It has G1, DNA synthesis, G2, and then M phase. All of this is interphase. And this is mitosis. Or meiosis, but right now we're talking about mitosis. You end up with two cells. You start out with one cell, you end up with two cells. I think that's basically what we need to know. What I really want you to know, the most important, is what's going on with the chromosomes. So there's a chromosome. Let's keep it simple right now. There's a chromosome, one pair of homologous chromosomes. Yeah, let's make it two pairs. Alright, sounds good. We do S phase, and then they look like this. So they're kind of spread out. They haven't lined up yet. They really don't look like this right after S phase. They start to look like that during mitosis. And then in mitosis, they line up. And then they divide. And when they divide, they kind of go back to looking like this. Well, these chromosomes are starting to look better. After 20 years of teaching genetics, at some point I should decide on a way to draw chromosomes. So that's kind of how they look like when they started. They went through S phase, and then duplicated, and then split and got back to the way they started. So if we toss some genes on there, let's say this is a heterozygous for two genes, then we duplicate these guys. Well, we'll come over here. We duplicate these guys. Big A. I'm going to put little b here. And we're going to get crazy. Put little a there. Put big B here. So where exactly they line up relative to each other on different chromosomes doesn't matter. What matters is that big A are on sister chromatids. Little b on the same sister chromatids. So then when we come down here, we have to make sure we get one of each. And over here, get one of each. The genotype of this guy starting out is this. The genotype of these guys is this. And the genotype of this guy is this. Because we have one of each gene. I know it's kind of scratchy, but the concept is start out one way, it's mitosis, so you end up identical. And so cytokinesis is a separation of the cytoplasm. The other thing I really care about that is sometimes we start talking about cells and we want to kind of know what the chromosomes are doing. And after cytokinesis is when the cells are completely divided. So we'll see cells sort of at this point like this. So they might be during anaphase, where the chromosomes are being pulled to either side of this cell. And then eventually they end up here after cytokinesis. But this cell here, it's still one cell. And so these chromosomes are considered to be in this cell, and these chromosomes are all in the same cell. So there are a couple problems where that came up. So what do we need to know about all this stuff? So these are the phases of mitosis. The chromosomes have undergone DNA replication, so they have 2 chromatids each. We'll just do this with 2 chromosomes so we have some room. The point here is they're all spread out. So in interphase, we'll just say G2. So G2 of interphase, the chromosomes are all spread out. You can't even sort of distinguish them as chromosomes. And then through this process, the chromosomes start to condense and line up. So I don't care how far along in the condensing process they are here or here, or precisely what all these things are. What I mainly care about is that they line up like this so here let's just do one, we're heterozygous for one gene, just for the sake of room. And then during anaphase, they look like this. And over here, they look like this. All the details of metaphase, prometaphase, all that nonsense, it just doesn't really matter. And then telophase, cytokinesis together, they end up in different cells. We just really care what's happening with the genes. So chromosomes spread out, chromosomes condense, line up, and separate. And in mitosis, the chromatids separate. Another thing that's sometimes confusing is that there are 2 chromosomes here. 1, 2. There are 2 chromosomes here. 1, 2. It's mitosis. So if you start out with 2 chromosomes, you have to end up with 2 chromosomes. If you start out with 100 chromosomes, you have to end up with 100 chromosomes, because mitosis ends up in identical daughter cells. Here's a lot of cool, I just like to keep this in there because there's cool pictures. So the DNA is stained blue. You can kind of see the DNA is all spread out. It's starting to condense in a little bit more. Down here, quite condensed and lined up. And you can see your spindles. Anaphase, the chromosomes are dividing. And then telophase, they're done divided. And then cytokinesis is just where you complete that division. That's all you need to know. Don't spend a whole lot of time talking about exactly when the nuclear membrane disappears, all that kind of nonsense. Look how these chromosomes are lined up. They're sort of single file. They're lined up in such a way that the chromatids can separate. And then the chromatids are in separate cells. So this particular type of cell has 2 pairs. So 4 total chromosomes. They line up in a row, and so we go from 4 chromosomes down to 4 chromosomes at the end. Even though these chromosomes look different from these chromosomes. These are chromosomes with 2 chromatids each. These are chromosomes with 1 chromatid each. They still are counted as a chromosome. So count the centromeres. I don't know why that always does that. I try to make a little circle. Little circle, little circle, little circle. 4 chromosomes, 4 chromosomes. Oh, there's a close-up. They are pretty cool pictures. You can kind of see the chromosomes starting to... Oh, there they are. That's cool. Ooh, they're all lined up now. Now they're dividing. We'll have to see some videos. Videos are cool. All right, so be able to draw chromosomes as they line up in mitosis. Very early question on the next exam. You'll notice that whenever I draw my sister chromatids are identical. It's because they start out like this. They go through S phase, which copies the sister chromatid. If you're going to copy something, it's going to be identical. Alright, we've got a little a, do S phase, copy it, you're identical. Now we all screw up from time to time and put a big A and a little a as different sister chromatids. And sometimes crossing over will cause that to happen. But that's why we ignore crossing over. Because I just want to drill it into your head that the sister chromatids have to be identical. Alright, consequences of the cell cycle. They're talking about the mitotic cell cycle. Genetically identical. That's it. Meiosis. Now we get to talk about sex. Buckle up, this is going to get exciting. Alright, production of haploid gametes. What does that mean? Go from 2N to 1N. We go from 46 chromosomes to 23 chromosomes. If you're a fruit fly, you go from 8 chromosomes to 4 chromosomes. You think I would know that? 8 chromosomes to 4 chromosomes. Why do we do that? Because then we're going to do fertilization. We take 2 of these haploid cells and go back to 8. So these are 2N, 1N, 1N, 1N. So 2 big consequences of meiosis. Reduction of the number of chromosomes. So a reductional division. And then genetic variation. And what do I mean by genetic variation? 2 things. Independent assortment. And, dang it, I said I was going to ignore crossing over, but we got to throw it in there. It happens. It's an important contributor to genetic variation. But the most important thing is that that's why meiosis occurs. That's why we have sex, is to generate genetic diversity. So meiosis has interphase, and so if you have interphase, you have chromosomes, and you have to go from 1 chromatid to 2 chromatids during S phase. So we need to get to this point because we're going to separate those chromosomes again. Then we have 2 divisions, meiosis 1, meiosis 2. This is the only one that really matters in terms of meiosis. So this is the point where we reduce the chromosomes, and this is where we generate genetic variation. So both of those things happen in meiosis 1. So here's the way it looks. Here's some chromosomes. They're all getting lined up here. It doesn't really show how they line up exactly, but it's showing that these are lining up in pairs. These are lining up in pairs. The red and the blue are like 1 chromosomes from mom, 1 chromosomes from dad. And then now what's going to happen is that we're going to divide in such a way that both of these guys go to 1 cell and both of these guys go to another cell. So we have 4 chromosomes here, we have 2 chromosomes here, we have 2 chromosomes here. So that's a reductional division because we've reduced the number of chromosomes by half. Then these guys are going to line up a lot like mitosis. And then they end up giving 2 chromosomes, each with 1 chromatid. So here we have 4 chromosomes, 2 chromatids each, 2 chromosomes, 2 chromatids each, 2 chromosomes, 1 chromatid each. So we go from 2N to N to N. So this is another equational division. There's only one reductional division in the universe. That's this one, meiosis 1. This is what gives us genetic diversity. This is what reduces the chromosome number. This is why we have sex. So you got a lot. You owe a lot to meiosis. So let's make sure we understand it. Clearly, this is something you need to memorize. Be able to draw this precisely. No, I still don't understand why they give you so much detail of this kind of stuff. But what do we need to know? We need to know that the chromosomes are kind of spread out. Then they start to condense and line up. So this is just like mitosis. The chromosomes have to condense, and then they line up in pairs. So this is what's going on here. You got your chromosomes. I'll draw them as X's, and they're lining up as pairs. So if you got a big A there, you've got a big A here, little a, maybe put a big B over here, little b over here. So they have to line up in pairs. They get kind of intertwined. This is where crossing over is happening, even though we're not going to talk about crossing over. But they start to get intertwined. This place where they get intertwined is called a chiasma or chiasmata. Crossing over occurs. The pairs start to. So this is anaphase 1, and the pairs are splitting. And so we still have 2 chromatids per chromosome, but these guys are splitting. And so we're not splitting the chromosomes in 2, we're taking full chromosomes and separating them into different cells. And then they have to decondense. So when we're all spread out and we go into a condensed form, that's called condensing. So condense the chromosome. So this would be decondensing. Just means they're scrunched up here. They just scrunch up so they can divide, and then they spread out. And then, where are we going from here? And then we go down and we start prophase 2, where they start to condense again. They condense and line up on the metaphase plate. Notice how this looks so much like mitosis. Now our chromatids separate. Then they spread out again. I didn't know they did that. I'm not sure they do that. I'm not sure that they actually spread out and then condense again. They're probably not condensed here. Even I guess they kind of condense, but that doesn't really matter. So, but notice what we have. We have 2 chromosomes per cell, 2 chromatids, so 2 chromosomes, and 2 chromatids. Whereas, so in metaphase 2, so these are in different cells. So each of these cells is undergoing division. So that has 2 chromosomes. That has 2 chromosomes. It's kind of in anaphase, so we'll see this is where it gets goofy. So technically that has 4 chromosomes because these chromosomes split in half. And then we have to count each half as a chromosome. But it doesn't really matter. Once they completely divide, we end up with 2 chromosomes per cell. So we start out with 4, and we end up with 2. That's what's important. The chromosomes look kind of cool. It's kind of nice. Look at these cells. Oh, look at how nicely lined up they are here. That's kind of nice. Oh, they're nice anaphase, getting pulled apart. Metaphase 1, getting pulled apart. Anaphase 1, getting pulled apart. Metaphase 2, they line up in a line like mitosis. And then they should get pulled apart in anaphase 2, sort of like mitosis. Then they separate into different cells. So consequences of meiosis. 4 cells are produced. Chromosome number in each cell is reduced by half. The new cells are haploid. And they're different from one another, from the parental cell. For example, we might start out with this, and then we'll get this. So meiosis is where this happens. So it's where you reshuffle these genes. And if you think of it this way, if we think of starting with 2 parents like this, where big A and big B are together, and little a and little b are together, and we make an F1, and then we do a dihybrid cross, and this gives us that. So where we started out with big A and big B only, and little b only, now we've got new combinations. We've got big A and big A and little b, this is a new combination. Little a, little b, this is a new combination because we didn't have that to begin with. So we've got some that look like the parents. We've got some that are a new combination. They're new kids on the block. Maybe a new boy band, new combination. All right. Blank screen, that means I should draw something. So let's draw meiosis, metaphase 1. Why? Because that's the most important thing in genetics. Draw them like this. This is my old school chromosome drawing. It's hard to draw them this way. Alright, big A, little a, little a. Sister chromatids identical. Let's put the capital letters on the left, little letters on the right. When these things divide, these are going to divide, then they're going to divide again, and we're going to end up with chromosomes that look like this: big A, big B. And this is a cell. We get 2 of those. And then we also get 2 with what's going on here. Alright, then we can also get little a, little b, little a, little b. These guys are going to be on chromosomes. And we're going to get 2 of these. And so we reduced. We went from big A, big B, little b, down to just little a, big B, just little a, little b. Alright, so that's groovy. That tells us how we're getting these 2 types of gametes. But how do we get the other kinds of gametes? Oh, look at these abstract fellas. Alright, big A, sister chromatids identical, little a, sister chromatids identical. Not only that, but homologous chromosomes need to line up in pairs. But now little b can go over here. Big B can go over here. After these guys divide, big A and little b are together, little a and big B are together. We can get this. We can get 2 of these, and 2 of these, and this is where we get all our gametes. So what did I just do? I explained how the chromosomes line up to go from that to this. Let's make that little a, little b, big A, little b, little a, big B, because of meiosis, in particular meiosis 1, and independent assortment. We don't need any crossing over to explain that. What kind of gametes would it produce if it lined up like this? Big A, big B. Oops, look what I did there. Look what I did. What's going on? No, that goes over here. Got quick. I almost messed that up. But big A, big B. Little a, little b. If they line up the other way with a big B over here and a little b over here, then we get big A, little b. Big B. Alright, I think you get it. Yikes! What's going on here? Alright, so this is showing you how you get genetic variation out of meiosis 1. So it's starting to make you think about, more than 2 chromosomes at a time. keeping in mind that we have 23 pairs of chromosomes in humans, and so there's lots of different combinations. And so what do we got here? We have 3 pairs of homologous chromosomes. Chromosome 1, chromosome 2, chromosome 3. The M stands for maternal. The P stands for paternal, meaning these came from dad and these came from mom. Undergo S phase, DNA replication. Now we have 2 chromatids per chromosome. And these guys are going to line up. So chromosome 1's always line up, chromosome 2's always line up, chromosome 3's always line up. This first alignment shows that all the maternal guys, all the maternal chromosomes are on the left, all the paternal chromosomes are on the right. But they can line up in any way. So here we've got the 3rd chromosome flipped, so the blue one's on the left and the red one's on the right. And down here we got 2 with the blue on the left and 1 with the red on the left. Lots of different combinations. So with 3 different chromosomes, so when we did 2 different chromosomes, we got 4 different things. If we have 3 different chromosomes, we get 8 different things. So the way you figure that out is if you're heterozygous, it depends on how many chromosomes or how many genes you're heterozygous for. And for each, and if we have big A, little a, we get 2 different things. So it's how many heterozygous pairs do we have? And there's a little formula that goes 2 to the n, where n is the heterozygous number of pairs. So 2 to the n. What's with all this math? I thought there wasn't going to be any math on this one. We had binomials, we had exponents with that, we got to multiply fractions. What's up with all this math? But n is the number of pairs of chromosomes. And then here, and then this gives us the number of different gametes. And the reason that we get all these different numbers of gametes is because the way the chromosomes can line up in lots of different ways. And so if we have number of pairs, number of different gametes, and so 2 to the n, so 2 to the 1 is 2. So for big A, little a, we can get big A and little a. 2, big A, big B, little b, 2 to the n, 4 different things. 3, 2 to the 3, 8 different things. 2 to the 4, 16 different things. 2 to the 23rd, so that's humans. That's a big number. 2 times 2 times 2, 23 times. It's somewhere on the order of 8 million. So these are number of gametes. And this is just like saying we have 1 gene per chromosome. So this one would be like this. And then this one would be all the way up to like Z, Y, X, W, W. So we have 23 pairs of chromosomes. 8 million types of gametes. That's a lot of gametes. But then you take that and you do a Punnett square. And that gives you the number of possible combinations of offspring. So you put 8 million gametes on this side, 8 million gametes on this side, because 8 million times 8 million is a lot of million. So this is 10 to the 6th, this is 10 to the 6th, This is 64 times 10 to the 12. So those are so when you have a baby with one person, you have the odds of getting a particular genotype is 1 in 10 to the 12. It's a lot of different combinations. So that really explains the uniqueness of every human. This is, sometimes people really get into exactly what's going on at each detail here. The only reason I show this is just to give you an idea to put the meiosis in context. So we're just going from 2n, meiosis, meiosis, meiosis. We get down to 1n. Same thing here, meiosis, meiosis, meiosis. You do get, there's some details with meiosis where you only get, you get these polar bodies. What we care about is fertilization. Make a zygote. Make a baby. Okay, there's a baby. And that baby is genetically unique because of all of the different combinations of genes that were contributed by mom and dad. All right, so that's meiosis and mitosis, being able to draw the chromosomes. Probably one of the most important things of genetics of this first section. It's very important. It'll be on the first quiz. It'll be on the first exam. It'll probably be on the second exam. When we start doing crossing over on the second exam, we're going to do crossing over and genetic mapping, and that's all meiosis. So meiosis, meiosis, meiosis. And it's all meiosis 1. We don't care about meiosis 2. So meiosis, we don't care about mitosis at this point. I mean, I'll have you draw the chromosomes in mitosis once, maybe, just to compare with meiosis, and then the rest is meiosis. And if you don't get it, come in and talk to me, and draw it, and keep drawing it until you get it. Because I don't, it's not like I'm going to surprise you and say, okay, the test is going to be about, I don't know. I don't even know what else it could be about. I mean, meiosis is the number one thing. So, have I said meiosis enough times? It's important to say, to understand meiosis. Me just saying it a lot of times isn't going to help. You looking at this video will help a little bit, but it's not going to be enough. You have to draw it out. Explain it to your friend. Please go to your friend and say, this is what I think is going on with meiosis, and see if they agree, and then have them draw it out for you. I think we could get there. I think all of us could draw out the chromosomes. All right, so that's it for chapter 2.